Good afternoon and good morning, everybody. So I have the pleasure today to present the speaker of this seminar. And I'm Giuseppe Stancanelli from EFSA, and I'm introducing now the speaker today, Alberto Fereres, who is a well-renowned vector entomologist from ICA-CSIC in Spain, who is leading the team in the Institute for Agricultural Research, working in ICSIC on vector plant pathogens. So this presentation will be about the research his group, together with other groups in Europe, are conducting on the American sharpshooter Draeculacephala robinsoni, which was found for the first time in Europe at the border between France and Spain in 2021. So I'll let the floor to Alberto and thank him very much for accepting to give this speech.
Thank you very much, Giuseppe, for the introduction. So I will go directly to the topic. Draeculacephala robinsoni, an American sharpshooter in Europe. Okay. So first of all, I would like to say that sharpshooters are very scarce in Europe. We have very few species compared to the Americas, and Draeculacephala robinsoni is one of the latest introduction. On the other hand, the spittlebugs, Aphrophoridae, are much more common, and as everybody knows now, Philenus spumarius is the main vector of Xylella fastidiosa in Europe. But we are having new introductions, and this is one of the most important introductions that we had from the sharpshooter side. So this species, Draeculacephala robinsoni, was introduced in Europe by 2021, and it was found in northeastern Spain and southern France. And it was the first time this species was recorded out of North America and Central America, because this is a species that has been only reported in the Americas. However, Draeculacephala genus is quite a big group, and there is a species which is called Draeculacephala minerva that has been reported in many different places, and there has been a lot of research efforts on minerva, not so much on robinsoni. Draeculacephala robinsoni is present right now in the Girona province in Spain, and it mainly appears on ryegrass, barley, alfalfa fields, and also in rice fields, as you will probably see later. I will be talking a little bit about systematics, and then I will be talking about the biology of the species, ecology, host range, feeding behavior, and transmission, a little bit of what we know about other species transmitting Xylella fastidiosa. And there has been no research at all on the vector ability of Draeculacephala robinsoni. So this is an empty space that we need to fill up. And then I will be finishing up talking about the likely role of Draeculacephala in the spread of Xylella in Europe. So first of all, this species has been reported, as I said before, in the Americas, but recently it has been found in northeastern Spain, and only 3 years ago it was found also in southern France. So you can see it's not only in North America, but it's also present in Central America. This is the distribution of the Draeculacephala genus in general. But Draeculacephala robinsoni is mainly present in the eastern North America, Canada and USA, at the east of the Rocky Mountains. So this is the distribution of robinsoni, while for Minerva, which is a very closely related species, it happens to be in southwestern USA and also in Central America. So as I said, this species was introduced in Spain and France, and it was reported in 2022. And this is the paper where they reported the presence of this insect as a new introduction. And as you can see here in the map, it's present in this area of southern Spain, southern France and northern Spain. this is called Girona province, okay? And here you can see the distribution of the species, mainly when it was first reported, this is the main hotspot, I would say, the place where it was found, Yeguada cenillosa, Coyure, and Lago de Sils, which is the places where it was found in 2021, 2022. But now the species has been moving northwards. Now it has been reported close to Montpellier in a place called Lattes, and also in Béziers. So it's present now in this area that you can see here. And also it has been moving a little bit southern to the province of Girona, touching the province of Barcelona, right? So this actually is the distribution of this introduced species at the present time. It's mainly here in the north of Girona province, close to the border with France, but it has moving down to the Punto de la Tordera, which is the southest place where it has been found. Intensive surveys have been conducted in all these green spots here, which is to the south of Catalonia, close to the Comunidad Valenciana. But there has been no reports at all yet from the species. So this is mainly the area of distribution at the present time in Catalonia. So this is the species. Here you can see a female, which is bigger and much longer than the male. This is the picture of a nymph, as you can see, which is a very distinct species that you can see the green color with specific taxonomic or systematic morphological characters that I will be describing now. Basically, this species is about 6.5 to 8 millimeters for males, or even almost 1 centimeter for females. The face is convex with a clippus being angulate. Also, it has a face which is of black color with a blue pigmentation on the pronotum, as you can see here. Also, another important fact is the genitalia, which is very close or almost identical to another species, which is called Draeculacephala portola, except for specific characteristics that have been described of the Areargus. These are the 3 species that are mostly related: Draeculacephala robinsoni, which is the one that we are talking today about, and this is the descriptions of the other very closely related species, Draeculacephala portola and Draeculacephala minerva. Especially Draeculacephala minerva is the one that we have most information on the transmission ability of the genus, and also on the biology and ecology. So we will be talking a little bit also about Draeculacephala minerva. So basically, the genus Draeculacephala comprises 3 major lineages that are well supported by synapomorphism, which means that there are 3 spots where they have been developed. One is related to Central and South America, which is the one of Clipeata. The other one is Draeculacephala crassicornis, which is mostly related to North Temperate America, North America. And then there is another big group of remaining species which has been concentrated in southeastern USA. So this is the cladogram with the basic connections of the species into those 3 major lineages. I would say that Draeculacephala robinsoni is very closely related to Portola, Minerva, and Producta, as it has been reported. And also, there are some synonyms. For example, Draeculacephala thea and Draeculacephala sphagmeticola are synonyms of Draeculacephala robinsoni. And by molecular analysis, and there is some analysis that suggests that Draeculacephala robinsoni and Draeculacephala sphagmeticola are a single species. And this same study reports that Draeculacephala thea, portola, and paludosa is a separate species. What we know about the ecology of robinsoni in the Americas is that this species is mainly present in the field from March to December, with a major peak in the summer months. And in the Americas, it has been reported that this species overwinters in the egg stage. It is not classified as an important pest in the USA, but in Central Valley of California, overwintering occurs in the egg stage, as I said before, and this has been reported as a character that may not impact too much its role as a vector. It might be not so big because infections may occur too late. And if infections are too late, there is no such a big impact on priest disease. But this, as I said, happens to be only in the USA, as we have no studies up to date in Europe. So we don't know the behavior it will have this species in Europe. Although I will give some information on what we have found in this last year of studies here. In the Americas, it's mainly present on grasses. And these are the species of grasses where it has been found. Phalaris, Theamais, Saccharum, Carex, Limos, mainly on grasses and also monocots. The life stage, as we have studied this life stage of this insect here in Spain, we have been doing some studies as part of an EFSA project that has started just 1 year ago. We have some information on the life cycle. One of the important things is that this insect can be reared and maintained on barley plants. And these pictures actually belong to some barley plants where you can find the eggs, which are placed here inside the stem, and they are not visible from the outside. They usually are between 6 to 12 eggs in each of the layers. Each time you can see them. And then there is another group of eggs here in mice, which you can see from the outside. So in the mice leaves, it's possible to detect or to observe the eggs which are placed in the cuticle of the leaf. So basically, in mice, we can see the eggs, but not in the other grasses. This is the life cycle as we have been rearing these species here in Madrid in our lab, and you can see these are some of the eggs that I explained. These are the small nymphs that eclode from the eggs. Then you have larger nymphs here, and then you have the adults. Basically, the cycle, although it's not very well known, under control conditions, it lasts about 45 days with 5 nymphal instars and with a 23-20 degrees centigrade in temperature, with a photoperiod of 14 to 10 hours. These 3 peaks that we can observe in summer, basically from May to late October, is what reflects probably the 3 to 4 generations per year that we have under our conditions. And there is no obligate diapause. So as we can see, the data that we have up to now suggests that the insect can overwinter in the nymphal stage or even as adults. So we can see adults in this time of the year, as well as nymphs in quite high densities, at least in the Girona region. So probably the absence of diapause means that this species doesn't behave the same as in the USA, because in the USA there is an obligate diapause, while here in Europe it seems to survive in the winter stage as adults or nymphs. The rearing conditions we have been setting up, which were the best conditions to maintain the insect, and we have found that light is very important. You need strong LED light to keep the colonies going. We tested several plants, species such as maize, lolium, oats, rice, barley, and the best one for us to keep the colony going was barley. Barley maintains a good number of individuals through the whole season. And we can keep this insect in barley throughout the year. We've been rearing the insect for about 1 year now. So this is the adult, how it looks like, the first instars, how it looks like, 14 stars. And also, as you can see, another important aspect is that these insect aggregates, they like to feed on the basal part close to the soil, and they prefer to feed on the stem of the barley plant. So you can see them mainly on the lower part of the plant. Draeculacephala minerva, we have much more information because it appears in the Central Valley of California, and it's very frequent, and it has also 3 peaks in the summer. Basically, in the winter time, as I said, this species Minerva also has a diapause, and also during winter time, the adults are very difficult to collect. So the egg diapause, where winter stage, at least in California for Minerva, it seems to be the egg stage. This insect is usually sampled when monitoring its populations by debug or by sweep net, and they are not very well responsive to yellow traps, but they are well attracted to UV lights. Minerva females like to stay on pasture, and they fly mainly after dark, mainly just after sunset. They have a peak of flight, usually 15 to 45 minutes after sunset, as reported by Sandy Purcell. Also, we do have some information on the population dynamics of these Minerva species, as you can see, and it also reflects the 3 peaks of adults very well during the summer time, 3 generations per year. In the case of Minerva, we do have a number of plants which are breeding hosts, mainly on the Graminia and Cyperacea family, but also in some other families. And feeding hosts, there are a large number of plants where you can find them. For example, alfalfa, Medicago is a good host, also Cyperaceas, and as I said, but many other species can be colonized or can serve as hosts for Minerva. This study reflects the preference for pastures and alfalfa, where you can see that the highest density was found in permanent covers, ground covers, in pastures and alfalfa in the study that was conducted in Talure County in 1977. Draeculacephala robinsoni in North America and Canada has been studied well. As I said, it's present at the eastern side of the Rocky Mountains. It's widespread not only in the USA, but also in Canada, in the west coast of British Columbia. Also, it can be, as I said, seen that it needs an obligate diapause because it overwinters in the egg stage. And there are a number of plants which have been recorded as host plants for this species, which you can see here in the list. In our case, we've been studying for the last year the presence and the population dynamics of robinsoni in different habitats, mainly in the Girona region. Basically, we've been sampling in avena, which is, oats, rice, perennial ryegrass, and alfalfa. Those are the major places where we have been sampling. It's present mainly in flooded areas, in humid areas, and what we have found is that they colonize rice fields quite commonly, and the highest populations we have recorded have been in rice so far. But it's also present in Lolium perenne, in perennial ryegrass, alfalfa, Paspalum disticum, and also in the ground vegetation of apple and olive orchards. In France, it has been reported in a series of Graminia plants and Cyperacea, but the species are mainly on the monocots, the major host plants. This is the place where we were sampling when the project started last year, 1 year ago, And what we happen to be is that there is a hotspot in a place called Senillosa Farm, where we find high densities of this insect. Jordi Sabater, who is part of the project, was informing us that that was the best place to find the insect in the winter, and that's what we did. We went to the sampling site, and we found a big number of adults and nymphs present 1 year ago. Basically, this was the first time we found the insect. We catch them and we took them to Madrid to start our rearing colony. This is just the time where all the team was able to make this survey and collect the insects for starting the project just in the kickoff meeting. On the other hand, we developed a sampling protocol to follow up the populations of this insect. Basically, here you can see the summary of what we decided to do. We were sampling in 10 different sites using in both ways of sampling, DBAC, which is reported to be the best way to collect adults, and also nymphs, and also we did some sweet net samplings to collect a number of samples within each of the 10 sites where the sampling was performed. This is the areas where the insect has been sampled in this last year, not only Catalonia, but also we've been sampling in regions close to Catalonia, such as Aragon, La Rioja, the Valencia community, as well as Galicia, which are also places where it could be found. But so far, there is no detection of robinsoni outside Catalonia, and there was no detection of Xylella fastidiosa in all of the insects that were trapped. So there is no presence of infective insects so far. The samples in Catalonia were restricted to these 4 areas where we've been collecting insects: Castelló de Empúries, Sant Pere Pescador, Aiguamolls d'Empordà, and Estany de Sils, which is here in the south. Basically, the place where we can find more insects, highest density, is this place, Senillosa Farm, where we sampled some grasses, pastures, perennial raygrass, and some oats with rice fields that were grown. This is a closer look of how it looked like in June 2025. This was lolium, woody edge, alfalfa, and then avena and rice, which was grown in this plot. I must say that this particular place where we can find high densities of this insect is very close to a place where they have a horse farm called the Eguada Senillosa, where they have a big amount of hay. So what we do not know if it's this hay that they could have been importing from abroad could be the source of introduction of this species. Of course, we don't have evidence that the eggs can survive in the hay, but this could be tested and this could be further investigated. But it's particularly important to note that there is a very high density of insects here, very close to the horse farm where they keep these horses in the Senillosa region. Basically, this is the population dynamics. So far, from January to July, we've been finding a number of captures in this region. Castelló de Empúries, for example, we have captured some insects in Perennium lolium, Lolium perenne. We have been capturing also in alfalfa here in the month of June. We have some in high populations when the rice was transplanted. In the rice, we can find large number of insects. Then we've been continuing sampling until now. Basically, we present here data until the end of October, where you can see also that rice is the species that has the highest population of Draeculacephala, followed by probably Lolium perenne. This is in terms of percentage. You can see that Lolium perenne is probably the best host during the year until we start to grow rice, which is the predominant crop in the summer, and the insects breed and move to rice just after August. This is in terms of percentage, the proportion of adults in blue and nymphs in red. So throughout the year, we have this distribution of adults and nymphs. And here we can see that by comparing the two sampling methods, SweetNet and debug, we can see that we can see that SweetNet can collect a high number of adults by the end of the season in October, while the debug is able to collect more nymphs than adults. So this purple line reflects the captures of nymphs, which seems to be collected more efficiently with debug than with SweetNet. So the samples the surveys that we've been doing not only restricts to Catalonia, but also to Galicia, Valencia, and Rioja, Aragon regions. We've been sampling these different, in each region, we've been sampling 10 sites, and sampling was performed every 3 months, looking to environments which were similar to the ones where we found the insect in Catalonia. But the Draeculacephala robinsoni has not been found so far. This is the places where we sampled in the Valencia region, mainly concentrated on rice crop, because we knew that rice was a good host for the insect. Also, we've been collecting making samples in Galicia, where we could find high populations of Cicadella viridis, but not Draeculacephala. And Rioja and Aragon, where we can also find Cicadella viridis, but we cannot find Draeculacephala. So these were the places where we did our sampling. Basically, these are some pictures of how the sites we're looking at, Valencia region, and also in rice fields, and also in vineyards in Galicia. Of course, we were looking to vineyards that had cover crops or that had grasses as ground covers. What about the feeding behavior? The feeding behavior of this insect is very specific and very difficult to conduct, because the insect prefers to feed on the lower part of the plant, but not only that. Once it stands on the plant, it has a particular behavior that it tries to cut the gold wire. As you know, the technique that we commonly use, which is called EPGs, electrical penetration graph technique, which gives you very good information on how the insect feeds on the plant, it requires a connection of the insect with a gold wire, which needs to be connected to the pronotum of the insect. But the insect tends to do a particular behavior with its legs, with its hind legs, and tries to cut the gold wire while it's standing or hanging from the gold wire. So we've been having some troubles to particularly connect the insects to the EPG recording system because of this particular behavior that it tends to cut the gold wire, if not properly managed. We found out that you have to first place the insect on a solid surface until it stays more or less resting, and then you can place it on the plant. But it's not easy, because sometimes it cuts the leaves, the gold wire. Also, We had to make a large Faraday cage, 2 meters high, because when we were using olive plants for conducting the EPG studies, of course, we need to do it with bigger plants, with woody plants that require some big Faraday cage, which we had to make. But now everything is working fine inside the Faraday cage, and we first started to do some EPGs on barley, rice, maize, and lolium, and recently we finished our recordings on olives. These were actually the first recordings we got from Draeculacephala, and we found basically all the different specific waveform patterns that we already recorded for other sharpshooters and spittlebugs in our lab. So right now we've been doing EPGs on those 5 different host plants, but we are planning to also do some feeding behavior studies with almond, grapevine, and alfalfa in the coming months. So what do we know about the transmission of Draeculacephala? Well, as I said, Draeculacephala robinsoni has not been studied as a vector, but as it's a silent feeder, it should be considered as a competent vector as any other silent feeder. But there is no specific studies on the transmission of Xylella fastidiosa Draeculacephala robinsoni, although there has been some studies that conducted with Minerva and other species which are related with robinsoni. The point here is that alfalfa could act as a host plant, which is a susceptible plant to Xylella fastidiosa. Also, grapevines and almonds are usually or many times associated with grasses or cover crops, where these species could build up high populations. And as I said, the species prefers to feed on grasses and also alfalfa. So any place where you have pastures or grasses, there might be high populations of Draeculacephala, which could be acting as vectors of Xylella. For Minerva, the highest infectivity of individuals transmitting Xylella were found in alfalfa fields. And the infectivity peak of these insects was found to be in October. Of course, this information refers only to Minerva. Pastures are reported as poor hosts of Xylella, but they are very good hosts for Minerva. So the insect could build up in high numbers in the pastures and then could acquire the bacteria at some point and transmit it to other Xylella susceptible crops. But the point here is that the few studies that have been conducting to understand the role of Draeculacephala as a vector of Xylella refers always to the ground covers under the grapevine plants. And usually they can find the insects in the ground covers, but inside the vineyards or on the vineyard canopy, the presence of this species is quite rare. So when there is this species present in a vineyard, for example, in a grapevine, transmission usually occurs only on the borders, okay? Because vine-to-vine transmission seems to be unlikely. This is what we know. And as I must say that all these studies have been conducted with Minerva, and nothing has been done with robinsoni so far. However, for almonds, it has been reported that Minerva can play an important role in transmitting Xylella fastidiosa in almonds and produce almond leaf scorch disease in the Central Valley of California. So what is the situation right now in Europe? Well, the situation in Europe now is quite dangerous because there is a big threat of having some important transmission of Xylella in case we don't stop or if we don't do any action against this species, because the species has been already reported in Lattes, which is very close to Montpellier, Béziers, which are two regions where Xylella fastidiosa is already present. So it's part of the demarcated area where Xylella fastidiosa, I think the multiplex species has been reported. So now we do have the new vector installed in this area, and this new vector could play some role in the transmission of Xylella in vineyards, which is basically probably the highest risk right now. And this is as I said, in this brown color, the place where the two pathogens or pests meet together. Okay, so Xylella fastidiosa demarcated area and Draeculacephala is already present in the demarcated area. This blue line spots the place where it has been found first in 2022. And the red spot or the red rectangle refers to the distribution, the present distribution of Draeculacephala robinsoni at the present time, which is from Punta de Tordera, which is in the border with Barcelona province, all throughout Girona and up to Montpellier. This is where all this species is present right now, according to the latest surveys. And this is the demarcated area from Montpellier to Narbonne and the south of Narbonne. Okay, so just to finish up, I think I am on time. I would like to make a summary of what I have explained today. It's important to note that Xylella fastidiosa has already been detected in regions where Draeculacephala robinsoni is currently present. This encounter might change the bacterial epidemiology scenario in Europe, leading to unpredictable results. We don't know what would be the role of this species as a vector of Xylella. Although some studies will be conducted in the future as part of the EFSA project. Also, we do know that grasses are commonly grown as ground covers, and these grasses that are ground covers could host a high number of these species, because Lolium and Brachypodium, for example, are common cover crops in olives and vineyards in Europe. We do have high population densities in rice, so this crop could be also hosting a large number of insects in areas where rice is grown. It could be important if we do have rice close to grapevines or other susceptible crops. It might be important to be aware that robinsoni could act as a vector. Also, it's important to know that cover crops, not only in rice, but in many other crops such as almonds, vineyards, or olives, all these cover crops, round cover crops, could be hosting a number of insects that could be involved mainly in primary spread of Xylella. In the case of Draeculacephala robinsoni, we still don't know if they can feed on vineyards, but we do know that it can feed on olives. So for short times, but we know now with our EPT studies that they are able to feed on olives in some cases. Of course, it's not a preferred host, but it can feed on olives if they are forced to. And then transmission of Xylella is a very fast process that can last only a few minutes. So even transient populations of robinsoni, if we don't see them in the canopies, for example, of olives or grapevines, just short visits could be enough to act as a vector. So there could be some transmission even if they do not colonize crops such as almonds or grapevines or olives in our situation. As I said, the other species, similar species as Draeculacephala minerva, do play a role as a vector in almonds, for example, in California. And finally, I would like to acknowledge, of course, the EFSA project about investigating the biology and capacity to transmit Xylella. which has been the major scope of this talk, and also all the members of the BioDrop project, which have been helping us working together on all these different aspects of the project, Belina, Jordi, Valerio, Marco, Daniele, and so forth. We have to acknowledge all their efforts of working all together and having very good success in completing the objectives of the project so far. And I also would like to acknowledge my team, of course, at ICA-CSIC, which are also part of the project. Rocío Lopez, who is the PhD student that is working on this project and conducting her PhD. And also Sandy Purcell, of course, for his valuable help on starting the project, who came to visit our lab, was able to start sampling with us last summer and help us a lot with starting the rearing colonies, working on the biology and life cycle of the insect. So thanks very much, Sandy, for your excellent help that you gave us. Thanks very much, and I'm open for questions.
Thank you very much, Alberto, for the excellent speech and very clear, and for all the work your group are doing.